A very warm welcome to the talk on early development of frog up to gastrulation. Frog belongs to class Amphibia, which represents a group of vertebrates that are transitional between water living and land dwelling forms. In frogs, the sexes are separate. The sexual dimorphism is well marked, that is, the male frog has copulatory pads on the palms of the forelimbs and also possess vocal cords for attracting female frogs for copulation during the breeding season. The frog becomes sexually mature by the first year of development. Its ovaries and testes get filled with ova and sperm respectively during the monsoon. Now, we will learn formation of ovum. The ovary of the frog is a flattened, multi-lobed structure with an inner membrane, the theca interna, and the outer membrane, the theca externa. In between these two layers are the primitive germ cells, blood vessels, pigments, connective tissue, some smooth muscle fibers and some fluid filled spaces. The primitive germ cells divide by mitosis to form many oogonia. The oogonia divide again by mitosis to form spherical masses of oogonial cells, which occupy different places between the theca interna and the theca externa. In such a group of oogonia, one oogonium enlarges to form an oocyte and the other oogonia become flattened follicle cells. Such a group of cells having an oocyte inside is called an ovarian follicle. The oocyte nucleus enlarges to form a germinal vesicle. In the ovarian follicle, the oocyte becomes further enlarged by deposition of yolk and some pigments in its cytoplasm. The yolk is in the form of microscopic plates called yolk platelets. The proteins of the yolk are synthesized in the liver of the frog and brought to the ovary by blood. The oocyte has a cell membrane, that is oolemma. Gradually, a non-cellular layer, that is zona pellucida, is formed between the cell membrane of the oocyte and the follicular cells. This layer is penetrated by microscopic finger-like projections, that is microvilli, from the cell membrane of the oocyte and also from the cell membranes of the follicle cells. As the oocyte develops further, the microvilli are withdrawn and the zona pellucida develops some fibers and is transformed into a membrane, the vitelline membrane or egg membrane. While the oocyte is developing, a layer of black pigment granules appears in the peripheral cytoplasm of the upper two-thirds of the oocyte. The pigment granules remain under the cell membrane, covering the contents of the oocyte. Each oocyte, later the ovum, looks like a black ball with its lower part white. The pigment is probably a metabolic byproduct of the oocyte. The yolk is heavily deposited only in the lower half. The less yolky upper half is called the animal hemisphere and the more yolky lower half is called the vegetal hemisphere. The number of chromosomes in the oocyte nucleus is 26, arranged as 13 pairs of homologous chromosomes. A little before ovulation, that is rupture of a follicle and release of the ovum, one member of each pair of chromosomes is thrown out of the oocyte as a small globular body, the first polar body. So, the oocyte now has only 13 chromosomes, that is half the number of chromosome of the species. Only after extrusion of the first polar body is the oocyte called an ovum. Moving ahead, we discuss the process of ovulation and spawning. When spring arrives, the ova are released from the ovary by the process known as ovulation. Ovulation is brought out by the secretion of gonadotropins from the pituitary. During this process, the ovarian follicle breaks and the ripe ovum is forced out through the epithelial covering of the ovary into the coelom. The ciliary action on the peritoneum serves to convey the egg to the mouth or infundibulum of the oviduct. This is also ciliated and the ovum is drawn into the duct. In about 2 hours after entering the infundibulum, the egg reaches the uterus, where it may remain for a day or two until this portion of the oviduct is full. The accumulated mass of ova is then expelled into water at the end of amplexus. This process is copulation. The whole egg mass forms a jelly-like irregularly shaped floatilla called the spawn, that is, a cluster of frog eggs which remain stuck together. On reaching water, the jelly absorbs water, begins to swell and lose much of their stickiness. Now, we detail the formation of sperm. Sperms are formed in the seminiferous tubules of the testes of male. From one primary spermatocyte having 26 chromosomes, 4 sperms are produced, each having 13 chromosomes. A spermatozoon of frog is about 0.03 mm long. It is flagellate type and is formed of head, middle piece and vibratile tail. The head contains a nucleus which occupies almost entire head and is produced anteriorly as acrosome protuberance. The middle piece contains 2 centrioles and several mitochondria. From the testes, the sperm first go to the vasa efferentia. From there, they enter the kidney tubules and come out of the cloacal opening. Thus, in the kidneys and beyond, they take the same path as that of urine. Further, we now discuss egg maturation and fertilization. The fully grown oocyte undergoes a series of major morphological changes in the process of hormone-induced maturation and subsequent insemination. Maturation requires 3 to 6 hours from the time of exposure to hormone and is accompanied by the disintegration of the nuclear membrane as the cell proceeds through the entire first meiotic cycle and arrests in metaphase of the second meiotic cycle. Fertilization is external, that is, it takes place in water. Only one sperm enters the egg through the animal pole. The sperm breaks through the egg membranes with the help of an enzyme hyaluronidase. The ovum forms a small projection, the cone of reception or the cone of fertilization. Through this, the sperm enters the egg. The sperm nucleus then moves towards the egg nucleus. The path of sperm nucleus is indicated by a streak of black pigment in the clear cytoplasm of the animal hemisphere. This path is called the penetration path. After the penetration of the egg, the sperm nucleus becomes invisible, that is, it cannot be stained by any known dye method. The middle piece and the tail of the sperm get completely absorbed into the cytoplasm of the egg. After the entry of the sperm, the vitelline membrane is separated from the egg plasma membrane and a perivitelline space is established in between the two membranes. The slight lifting of the vitelline membrane permits the egg to rotate freely within it, so that the heavier vegetal hemisphere comes to lie downward and the darker and lighter animal hemisphere comes upward. Such a rotation is called rotation of orientation or rotation of dorsoventral polarity. Along with rotation of the cytoplasm within the membrane, the cortical granules burst open into the perivitelline space and their contents adhere to the vitelline membrane, which now becomes tough fertilization membrane and checks the entry of other sperms into the egg. Meanwhile, the stimulus of the entrance of sperm inside the egg incites the completion of the second maturation division of the egg nucleus, which has postponed in the metaphase 2. It gives off second polar body at the animal pole and becomes vesicular to become egg pronucleus. Now the sperm nucleus also becomes vesicular and is called sperm pronucleus. The sperm pronucleus along with its centriole move towards the egg pronucleus and ultimately both pronuclei fuse together to form zygotic nucleus. At this stage, the zygote nucleus can be stained and seen. The zygote nucleus contains the diploid number of chromosomes. The next topic is cleavage and blastulation. The cleavage of a fertilized and telolacethal frog egg is radially symmetrical and holoblastic. The frog egg contains much yolk which is concentrated in the vegetal hemisphere that acts as an impediment to cleavage. Thus, the first division is meridional and begins at the animal pole and slowly extends down into the vegetal region. The cleavage furrow bisects the grey crescent, that is a structure formed on the opposite side of sperm entry in a frog egg, and then slows down as it approaches the vegetal pole. While the first cleavage furrow is still trying to cleave the yolky cytoplasm of the vegetal hemisphere, the second cleavage has already started near the animal pole. This cleavage is at right angles to the first one and is also meridional. The third cleavage is equatorial. However, because of the vegetally placed yolk, this cleavage furrow is a little above the equator region of the egg, that is, occurs in the animal pole region. This therefore is a ring-like cleavage arising separately in all the 4 blastomeres. It divides the frog embryo into 4 small animal blastomeres, that is micromeres, and 4 large blastomeres, that is macromeres, in vegetal region. This unequal holoblastic cleavage establishes 2 major embryonic regions, a rapidly dividing region of micromeres and a more slowly dividing macromeres area. As cleavage progresses, the animal region becomes packed with numerous small cells, while the vegetal region contains only a relatively small number of large yolk-laden macromeres. In amphibians, the embryos containing 16 to 64 cells are commonly called morula. At the 128 cell stage, the blastocele becomes apparent and the embryo is considered to be a blastula. The first cleavage furrow widens in the animal hemisphere to create a small intercellular cavity which is sealed off from the outside by tight intercellular junctions. This cavity during subsequent cleavages enlarges to become the blastocele. As the cleavage proceeds, the addition of blastomeres to each other increases and they arrange themselves into a true epithelium called blastoderm. This blastoderm remains 2 cell thick towards the animal pole of the egg, and here it forms a sort of roof of the blastocele. The sides and floor of the blastocele are occupied by multilayered blastoderm of large yolky macromeres. The resultant embryo, having fluid-filled blastocele and blastoderm, is the blastula of frog. Lastly, we will learn fate maps. In the blastula, the blastomeres, which have to form different germinal layers and different organs of adult frog, have their representation at the external surface of blastula. The fate of each type of blastomere has been observed by artificial vital staining methods of Vogt in 1929, and prospective organ region maps or fate maps have been prepared for the blastula of frog. The entire pigmented area on and around the animal pole, the prospective ectoderm, consists of two main areas. The epidermal ectoderm, which is to become the skin epidermis, occupies the anteroventral side of the blastula, and the neural ectoderm, along the future dorsal side of the embryo, develops into the central nervous system. The material for the sense organs is also contained in these two areas. Inside the neural ectoderm area occurs a small sub-area which develops into the eyes of the embryo. Similarly, inside the epidermal ectoderm occurs the material for the nose, ears, and ectodermal part of the mouth. An intermediate crescent area of grey color is the marginal zone of perspective notochord and mesoderm around the equator of the blastula. The material for notochord occupies a large area on the dorsal side of the blastula. Beneath the notochordal area and nearer to the vegetal pole is a narrow strip of precaudal plate. The parts of the marginal zone on both sides of the notochordal area are taken up by the material for the segmental muscles. At the upper side of notochordal area and towards the animal hemisphere lies another narrow strip of cells which develops into the tail mesoderm. The lateral and ventral parts of grey crescent give rise to the mesodermal lining of the body cavity, the kidneys, etc. The area on and around the vegetal pole is the prospective endoderm, which forms more or less non-pigmented area. Just beneath the precaudal plate lies the material for the anterior part of the alimentary canal, that is, the endodermal lining of mouth, gill region and pharynx. The remaining endodermal area includes the material for midgut, hindgut, liver, pancreas, urinary bladder and certain endocrine glands. Gilchrist in 1968 has named the zone of prospective ectoderm as zone of expansion because during gastrulation this zone expands downward and then converges toward the vegetal pole. Marginal zone of grey crescent has been named as the zone of involution because during gastrulation it rolls round and beneath the lips of the blastopore. The prospective endoderm has been named as zone of invagination because during gastrulation it sinks and glides into the blastula. At the end, we summarize the talk. The frog has been historically used as an amphibian animal model of development due to the ease of observation from the fertilized egg through to tadpole stage. The later metamorphosis of the tadpole to frog has also been studied for hormonal controls and limb development. Soon after the fertilization, the egg begins to proceed from a single cell stage to a many-celled embryo by a process of division or cleavage. Cleavage in frog embryos is holoblastic and radially symmetrical due to large amount of yolk present. This yolk, which is concentrated in the vegetal hemisphere, is an impediment to cleavage. Thus, the first cleavage begins at the animal pole and slowly extends down into the vegetal pole, dividing the egg into two blastomeres. The second cleavage furrow is also meridional, but at right angles to the first, dividing the egg into four blastomeres. The third furrow is at right angles to the first two and separates four upper small pigmented micromeres from the four lower yolk containing megameres, and the embryo now has 8 blastomeres. A single celled egg is thus converted into many celled embryo, which at this stage is known as blastula. A series of changes converting the single layered blastula into a two-layered embryo or gastrula are collectively known as gastrulation. Thank you.